All right, good morning, everybody. This is David. We're going to go ahead and get started. I want to thank everybody for being here this morning for our December installment in our science communication webinar series, Understanding Our Wildlife. And today we're very fortunate to have Heather Evans. Heather is our agency's conservation geneticist. And today, Heather's presentation is going to be on genetic applications for conservation and management. So again, thanks for everybody being here, and thank you, Heather, for being willing to give this webinar, and I'll turn it over to you.
All right, thanks, David. Yeah, so I am excited to be able to give this presentation today. I have been at the agency now for a little bit over a year, and when I first came in, I was really impressed with the amount and breadth of genetic work that was already going on. And so one of the things that I hope to be able to accomplish in this position is facilitating sharing the genetic work that's going on that everyone is working on, so that people can think about if what one group is using genetic applications for might also be useful in their system or species of interest. And then the other thing that I want to do is just kind of help demystify the genetic process so that it doesn't seem like you guys send samples off to whoever's doing your genetic work and it's a black box and then it spits out data. But helping to really illuminate the process and the methodology and also the caveats that come along with genetic applications. So when I talk about conservation genetics, I am really thinking of, there we go, a broad array of tools that can be used to answer ecological questions. And that can be things like using genetic markers to look at population structure. So how many populations do you have? What are the boundaries of those populations? What is the genetic diversity of those populations? How are they connected to each other? And then you can also use genetic markers to look at issues of hybridization, parentage and relatedness, population estimates. Then there are other tools that we can use, eDNA, so looking for a presence absence of species that may be difficult to find using traditional survey methods. You can use sequencing to look at species identification, or taxonomic studies, and high throughput sequencing to look at questions in diet or health. So lots of different areas where genetics may be able to help answer your research questions. So what I'm going to do today is focus on just a couple of these genetic tools and dive a little bit more into the methodology and start to illuminate that a little bit more for you. So what we're going to talk about today, the first one we're going to talk about is parentage-based tagging. And in a nutshell, this is when we compare genetic profiles of offspring to parents. We've been doing genetic parentage-based tagging on shad since 2010 now. And a little bit of background on American shad. I'm getting a lot of feedback. Oh, there we go. Thank you. So a little bit of background on American shad. This is an anadromous fish. They come into our rivers to spawn, then head out into the ocean where they spend most of their adult life, but they come back into the rivers at starting about age 3 to spawn. So this is a graph, you're looking at the green bars here of commercial landings for American shad from 1950 to 2016. And obviously there's a really precipitous decline in the commercial landings that we've seen since then. In the 1950s, they regularly caught 8 to 10 million pounds of shad. And then in the last decade, it's been consistently under 1 million pounds. So to combat this decline in population, we in CWRC started stocking American shad fry in 1998. That obviously represents a tremendous monetary fund commitment as well as time commitment, and hatchery managers needed a way to track the fish that were being put into the river. Were they surviving? Were they coming back to contribute as adults? So originally they tracked these using OTC methods, oxytetracycline, and then did a study about a decade ago and found that genetic tagging was more reliable than OTC markings. So we switched to genetic parentage-based tagging in 2010. So I'm just going to run through the methodology a little bit. So this starts again in the spring when these fish are coming into the river to spawn. Our biologists go out. The data that I'm going to talk about today is specifically looking at the Roanoke River. So go out into the Roanoke River and collect these fish, where they are transported to the hatchery and spawned in these big circular tanks. Each tank will usually get about 80 brood fish, and we have 4 tanks usually, so we're talking about 300 to 400 fish that are used for breeding. Because we have every fish that's used for breeding, we can then do this genetic parentage-based tagging, where the biologist will take a fin clip from each of the parents and send it to me. So this is what I get. I get this vial with the label inside, with the sample number and a small piece of fin clip. I'm going to use about half of that, what's shown in the picture, in order to do the DNA extraction. And DNA extractions are really just basically adding some soap and some enzymes to break apart the cells and release the DNA. We clean it with ethanol and make it suitable for downstream reactions. So in this case, we're going to do polymerase chain reaction, or PCR, to amplify specific sites throughout the genome that will help us to carry out this parentage-based tagging. And the markers that we're using for this particular study are microsatellites. So microsatellites are short segments of DNA that contain a repeat sequence. They generally occur in non-coding areas of the genome, and because of that repeat, they're unstable, such that during DNA replication, an additional repeat is often added. So this leads to a really high number of alleles and a lot of variability, and then power to be able to distinguish individual fish from each other. Most of my alleles or markers that I use have at least 20 different alleles. The way that we call microsatellites is based on size fragments. So we're really just looking at how many base pairs is in this fragment that we're amplifying. We'll get a primer sequence and we'll lay it in the flanking region of the repeat, do PCR, and then look at how large that fragment size is. So in this marker, we have one allele here that after amplification is 284 base pairs. And then our second allele has an additional repeat, this ACAG. And so it's going to be 4 base pairs longer than allele 1. So this is how we call it. We'd call it 284 and 288 for the alleles. And we compile this genetic profile for each fish. For American shad, we're using 12 microsatellite markers. These are the names of the 12 microsatellite markers, and here you see, so for marker CO10, the first allele is 301, the second allele is 313, and then in marker D021, we have a fish that's homozygous for this 267 allele. And once you compile genetic profiles for all your fish of interest, including the broodfish and any offspring you want to test, you can then compare those profiles to each other and start answering some of your questions. So if you had just a handful of fish, you could do this by hand. And so that's what we're going to do now. This is actually taken from an activity that we have at the museum. Whenever we're doing a study at the museum, we try to come up with an education cart that will illustrate something about the genetic techniques that are being used, as well as something about the species or system of interest. And then our volunteers and our interns take that out into the public side of the glass wall that we're on, where they can present the material to the public and help teach them about what we're studying and what we're working on behind the glass. So I mentioned this, if any of you have a project that has a genetic component and would like to have an education cart, please reach out to me. We can develop one of those. So in this particular example, we simplify it a little bit. So here we have an offspring profile. We're going to use 4 markers for this exercise, and we give them the mother. And then the point of this exercise is to determine who the father is, which of these 3 fathers, if any, is the father of this offspring. We call this who's the shaddy. And so we'll just go through this exercise. Here in marker 1 for the offspring, we see that there's a 136 allele and a 140 allele, and that 140 allele was inherited from the mother. And so what we're looking for from the father is a 136 allele. Father 1 and Father 2 both have that 136 allele, while Father 3 does not, so we can eliminate him as the father. Looking at marker 2 then, in the offspring, we see that the mother was homozygous for 214, so she had to contribute that 214 allele. We're looking for a 218 from the father. Both Father 1 and Father 2 have that 218 allele, so they're still both in the running. And moving on to the third marker, we're looking for a 271 allele from the father. Father 1 does not have that 271 allele, but father 2 does. So just to round it out, we'll look at that fourth marker and see that yes, the father could have contributed that 301 allele to the offspring. So father 2 then would be our Shaddy. And of course, we don't have just a handful of alleles when we're doing this. For our study, we have thousands of fish, and so we let the computer program do this kind of analysis for us. We use a program called Service, and this is kind of a typical output for the service results. It's using a maximum likelihood analysis, and where it will tell us what the most likely parent is for any given offspring, and then give us a statistical score that tells us what the likelihood is that that is the true parent. So if we look here, we have 5201 as the first offspring, and it says the most likely parent is this M406. But the statistical likelihood of that is negative. The LOD score, the logarithm of the odd score, is negative 27.1, meaning this parent didn't actually match any better than many other fish in the pool. And so we wouldn't consider this a match. It didn't match to any of our hatchery fish, and so by default it was spawned in the wild. Compare that to 5203, where it tells us that the most likely parent is 2031 with a positive LOD score of 19.4. So for reference, a LOD score of 3 would indicate 1,000 to 1 odds that the linkage observed did not occur by chance. So that's pretty high. And then it tells us that the second most likely pair, because we want to get the whole family, is this fish 2055 with a LOD score of 17.6. And then it takes all 3 individuals that have been called, puts them together in one family group, and gives us a trio LOD score. What are the odds that this trio constitutes a complete pairing? And here we see a trio LOD score of 41.2. So we would call this a hatchery fish. And then I have the data, so I can go back then and I always double check, is this a male? Is this a female? Did they spawn in the same tank? So that's just kind of a nice check that I have to be able to make sure that everything was input correctly, that the genetic analysis is working. In this particular example, then at the end, we have 3 fish that were identified from the hatchery and 3 that were not. And so we have a 50% hatchery contribution. And that's really the primary question here is what percent of the river of our American shad population is coming directly from the hatchery? So now that I've walked through the methodology a little bit, I'll show you guys some real data. So this is looking at percent hatchery contribution for juveniles in the Roanoke River. So they go, after going in the spring and collecting the broodfish, they'll go back in the fall, collect juveniles, collect fin clips from the juveniles. We then do the same genetic profile construction using the same markers, and then compare those genotypes to the broodfish genotypes and do the analysis that I just described. So here in the brown line, we have the percent hatchery contribution for juveniles, and this green line is the number of fry stocked in millions every year. And one thing that I want to point out is that the number of fry stocked won't necessarily directly translate to your percent hatchery contribution. So in 2012, we stocked just as many fish as in 2011 or 2013, but for whatever reason, the fish that year did not do well, and we had a really low hatchery contribution that year, compared to fish stocked in 2011 or 2014, where we had really high hatchery percent contributions of 42 to 45%. Now, so we look at percent hatchery contribution for the juveniles, but we can also look at percent hatchery contribution of those returning adults that are coming into spawn. So again, we started genetic parentage-based tagging in 2010. Shad come in force really starting around age 3. And so the first year that we picked up any of our fish that were genetically tagged was in 2012. So we had a couple of 2-year-olds. It's a little bit early, but we did pick up a couple. And then in 2013, you see this really start to pick up. We had about 8%. And because we're doing genetic tagging, we can tell you not only if it came from the hatchery, but we can tell you exactly what year it was spawned, because we can tell you exactly who the parents were. So as we continue throughout the years and add age classes, our percent hatchery contribution for returning adults increases. 2018 was the first year that we saw that 2010 breeding class drop out. So we think 2017 is probably the first year that we have a full complement of age class structure that's contributing to the spawn that we're able to pick up genetically. And so if we look at percent hatchery contribution in 2017 and 2018 and 2019, we're hovering right around 65 to 70% hatchery contribution for those returning adults, which is pretty high. So the nice thing about doing this kind of analysis is that once we have these microsatellite markers, we can actually run several different analyses. So this particular analysis was run with Service to look at the parentage-based tagging, but we can take the same exact data set and run it through some other analyses to look at other population genetics. So we can look at things like genetic diversity, population structure, the effective population size. And so this graph is doing just that. We took the same data set for the adults and ran it through a different program to calculate effective population size. Just as a reminder, the effective population size isn't measuring the actual number of fish in the system. It's really a measure of how many distinct breeders you might have in your system. And what we see is that from 2010 to 2017, we saw a decline in our effective population size. For the past several years, it's been hovering kind of right around or even just under 500. And so that's lower than we would like it to be. We also took the same data and ran it through structure. So this is a bar graph looking at structure, and every line here is an individual fish. So this is a fish and this is a fish. And then the red color indicates the probability that it belongs to population 1, and the green color indicates the probability that it belongs to population 2. And what we see in this 2016 analysis is that all of our fish taken from the Roanoke and the Neuse look similar to each other. They are consisting of one genetic stock, whereas fish pulled from the Petey River constitute a second genetic stock. But what we see in the Roanoke and the Neuse here is that it's pretty uniform. Compare that to this 2018 structure bar plot. So this is just looking at Roanoke, and what I'm seeing is structuring happening within the river. So that was interesting. Because every line again is an individual fish, I can go back to my data, I can match the line with the individual fish and figure out who are these fish that are constituting a second population. It turns out every single one of those red lines came from one of 2 parents that were used at the hatchery to breed fish. So in essence, we've created a founder's effect. And we've seen this in a few years now, where even though we are bringing in 300 to 400 fish to use for breeding and spawning, there's always a handful of fish that are contributing the majority of our spawn. And so it's creating this kind of founder's effect. So you have a few fish really contributing the majority of your spawn every year, and then 65% of your returning adults come from those hatchery spawns. And we think that is why we're seeing that decrease in effective population size. So this data was considered in making a decision in 2019 to halt stocking in the Roanoke. We were taking a 3-year pause. So we didn't stock in 2019, 2020, or this year in 2021. We won't really see the effects of that until 2022 when those age 3 class fish start to come back in. But we're hoping that taking a pause might allow for some additional wild spawning to occur that would increase the genetic diversity levels. We continue to monitor them every year. So we continue to monitor by electrofishing and genetic analysis, and we're just keeping an eye on it. So I'll take a quick moment to mention that this study was done with microsatellites. The same kind of study can be done using another type of genetic marker called SNPs, or single nucleotide polymorphisms. There are advantages and disadvantages to both systems depending on what you're doing, what your source sample is, and what your overall question is, but most of the programs that I use for population genetics will take either SNPs or microsatellites. I just have to tell it which one that I'm using. And then to give you kind of a quick overview of the breadth of genetic work that's currently going on, this is a list of some either recent projects or ongoing projects or currently proposed projects. So using genetic markers, of course, the American Shad parentage-based tagging that I just talked about. We're also currently looking at doing a Lepomis parentage-based tagging that would actually be for 4 different species of Lepomis. We use SNPs to look at black bass hybridization. We have a current study looking at urban deer density and population estimates using microsatellites. Another proposed study looking at cottontail landscapes genetics and hybridization that would use SNPs. We recently received a grant from NCDOT to use genetic markers and look at mussel population structure and parentage-based tagging. And then there's others that I'm not involved in at all, but I wanted to mention because I know they're going on and it's great work. So Lori Williams, who recently used SNPs as part of a new salamander species description, Matthew Godfrey, who uses microsatellites to look at sea turtle population structure and nesting fidelity, and Jeff Hall, who's used microsatellites to look at gopher frog population structure and inbreeding. So lots of different species, lots of different systems, and ways that these genetic markers can help answer ecological questions. So I'm going to segue now. I'm going to leave genetic markers and talk about a different technique that we use to answer some of these questions, and that's called metabarcoding. So a lot of people might be familiar with barcoding. So barcoding is when you have a single animal, you can extract DNA and then use a particular gene. A common one that people use in animals is called CO1. And it's capable of giving you a species identification because the CO1 gene has evolved rapidly. It's basically kept up with species evolution. So, and when you do these kind of barcoding analyses, you're looking at DNA fragment sizes of around 600 to 700 base pairs. We can process 24 samples at a time on our ABI 3500 that's in the lab. We also use that machine for the fragment analysis that I just mentioned previously. So metabarcoding then is the same kind of technique, but instead of looking at a single sample at a time, where you can look at multiple samples at the same time and process them in parallel, and you get shorter reads. The reads are usually about 150 to 300 base pairs long, but you get millions of reads at the end. So this kind of technique can be used with things like eDNA or diet studies or health studies. So for eDNA, we talked a little bit at the beginning about how we can use eDNA to look for presence absence of a particular species, but you can also use eDNA to basically sequence anything that's in your sample, and anything and everything. So I recently talked to one of the museum curators about the possibility of using eDNA as a way to bank snapshots of organisms that are living in a particular ecosystem, because as climate change continues to occur and anthropogenic activities continue to affect our wildlife, we have seen and expect to continue to see changes in the community structures of our wildlife ecosystems. So one possibility is to kind of take samples of eDNA, filter the DNA out, and then have them banked at museums so that we have specific time shots and time frames of what's living in our ecosystems at any given time. Another application for metabarcoding is diet studies. This graph is taken from a paper I published earlier this year where we compared a morphology-based diet study to a high-throughput sequencing using metabarcoding. What we found is that in the morphology-based diets, we're identifying tens of species, and using metabarcoding, we're identifying hundreds of species. And the last one that I'll mention today is animal health, and I'm going to talk about this one in a little bit more detail. So we currently have a project going on with our Tar River spiny mussels to look at animal health. These Tar River spiny mussels are federally endangered. They're endemic to North Carolina, occurring in the Tar and the Neuse River. And whenever currently our biologists find a Tar River spiny mussel, they bring it back to the hatchery to use for breeding in our augmentation. And we had an issue this year where these brood mussels became sick. We didn't know why. There were some black deposits noted on some of the shells, and so our current hypothesis is that there's some kind of bacterial or fungal infection that's impacting our brood mussels. And so we're going to use metabarcoding to look at that. We're going to amplify specific genes 16S for bacterial and 18S for fungal, and look at healthy and unhealthy muscles and see if we can find something that might be indicative of what's going on with our muscle health. So the way that we did this is we took swabs. We swabbed 3 different sites on the muscle, the foot, the mantle, and the visceral mass. We chose these 3 sites because they can be swabbed non-invasively without killing the animal, which is obviously important for an endangered species. A lot of previous studies have used gut, but you can't get the gut without killing the animal. So of necessity, we're looking at some microbiome profiles of different sites in the animal that haven't been looked up before. And then we had 8 healthy wild tahr river spiny mussels, 3 unhealthy tahr river spiny mussels, and then we also had tanks of propagated mussels. We had 1 tank that was doing fairly well, another tank that was not doing well, and so we used these propagated mussels to augment our sampling and took 10 from each of those tanks. And so again, we swabbed those 3 sites from all the samples mentioned, extract DNA then from the swabs, and then we PCR amplify for genes this time, either, as I mentioned, 16S, which is going to be bacterial specific, and then we used 18S to amplify fungal species. But 18S will also amplify really any eukaryote, but we're also interested in microorganisms that might be affecting muscle health. When we do these PCR amplifications per sample, we're adding unique indices to those primer sets that then get incorporated into the PCR product, so that after we put it on our high throughput sequencing machine, when the sequences come off, we can say, okay, these 100 sequences match sample 1, these 100 sequences match sample 2, and so on. This was run on a MiSeq, and when you're doing high throughput sequencing, you can there's different levels of machines. So the MiSeq is the lowest one. It will give you 20 to 25 million sequencing reads per run. If we want to run anything higher than that, we can take it to NC State and their core facility, where you can get 400 million sequences per run or 800 million, all the way up to 2.5 billion sequences per run. So really just depending on your needs. For this study, we were able to use the MiSeq in the lab. And so I'll show you some of that data now. So this is a PCOA plot looking at 16S, so bacterial profiles. And the first thing that I looked at was, is there differences between the 3 swab sites? Are there differences between the foot and the mantle and the visceral mass? In this PCOA plot, you're looking at clustering. The more closely items are clustered together relative to each other, the more alike they are. This was done with a weighted unifrac, which takes into account relatedness between taxa that are being identified. And what you can see here is that there's some separation here between this green, which is the visceral mass, and the blue, which is the mantle. We'll kind of point that out here. And then it can be hard to tell when you're looking at these PCOA plots, because this is really plotted in 3D space, but we're looking at it in 2D. So we always do a pairwise comparison, which will give us a statistical significance about if what we're seeing is real and if it's significant. So here we see that the mantle and the visceral mass have a Q-score when we do this pairwise comparison of 0.003. We consider anything less than 0.05 to be statistically significant. And so we are seeing that there is a difference in the bacterial profiles between sites on the muscle itself. And of course, that's interesting, but what we really want to know is, are there differences between the healthy and the unhealthy groups? And so that's the next analysis that I'll show you here. And we see pretty strong clustering here. So in green, we have our wild unhealthy. This orange is our wild healthy. The blue is propagated unhealthy, and the red is propagated healthy. And we expected there to be differences between our healthy and our unhealthy groups. What we didn't expect was to see differences between our propagated groups and our wild groups. I'm calling them, they're wild, they were collected from the wild, but they weren't collected this year. Some of these animals had been at the hatchery since 2009. And despite being in the hatchery for over a decade and having the same water source, the same filtrations, the same nutrients, they are maintaining a separate bacterial profile. So that is interesting. And then, of course, the question is, well, what is it exactly that's different between these groups? So this is a taxonomic bar plot of our 4 groups. Again, so propagated healthy, propagated unhealthy, wild healthy, and wild unhealthy. And one thing I'll point out here is the levels of diversity that we see. So our propagated groups are more diverse overall than our wild groups, and our wild unhealthy group is the least diverse. So we are definitely losing diversity in the unhealthy wild, at least, versus the wild healthy. Okay, so what are these? Each color represents a different taxa. And when I ran an analysis called an ANCOM analysis that measures differentially abundant taxa between groups and found 217 differentially abundant taxa. This project is currently ongoing, and so that's where I am now, is working through those differentially abundant taxa and trying to figure out what they are, what might be happening in with the muscle health, and if they're might be important. So I'll just point out a few right now. This one is interesting. It's a bacteria called Neisseria, and it's interesting because this is one we only see in our wild groups and not in our propagated groups. One of the challenges in working with bacteria is that there's a lot of bacterial species out there, and actually not a lot of studies on all the full complement of bacterial species that exist. A few have been studied well. They're usually ones that are involved in human pathogen activity, but of course we know that things that may be pathogenic in humans don't necessarily translate to pathogenicity in mussels, and vice versa. So all of that, which is to say, I don't know a lot about the things that I'm finding. So this is going to take some really deep dives into the literature searches, and sometimes there's just not anything to find. So this will be a project that will continue. As I mentioned before, this NCDOT muscle grant, part of that will be looking at muscle health, where we can continue to explore these questions. Some other things that I found were specific to only the wild unhealthy mussels. So this Hydrogenophaga, Chlorobi, and this Acetominobacter. And then again, I can't tell you much about them. The Chlorobi, I can tell you, is a green sulfur bacteria that's typically found in freshwater where light is limited. So that's, at least we know something about it and might be indicative. And then the last one I'll mention is this one. It's a disulfovibrio. And this one is interesting because it's one that's found in both of our unhealthy groups. So I mentioned those last 3, which we did see specifically in the wild unhealthy, but if something is truly pathogenic, I would expect it to be found in both our propagated unhealthy and our wild unhealthy. And this disulfovibrio fits that expectation. I mentioned at the beginning that we looked at 18S as well as 16S, and so here's an instance I'm going to point out one of the caveats or things that you should be aware of when looking at high throughput sequencing. So this is a table showing this column is average raw reads per sample. So coming off the machine, 16S had about 42,000 raw reads per sample, and 18S 33,000. Those are both good numbers. Then we filter them, remove low quality reads and negatives. We had 24,000 for 16S and 20,000 for 18S. And then we look at the total number of features. So a feature is how many unique, distinct sequencing reads do we have? Because in this 24,000, some of those are going to be the same thing sequenced multiple times. But for 16S, we actually had 21,000 unique reads, so 21,000 features. 18S, we have 2,000 total features. That's not unexpected. 16S is a more quickly evolving gene, so it can identify things to species level. 18S evolves at a slower rate, and so it really only identifies things to family level. So for example, Tarver spiny mussel has an 18S sequence, but it shares that same sequence with every other Unionidae. So the Tarver spiny mussel will look the same as a Lictio, will look the same as a Velosa. All of those will have the same 18S sequence. So that's fine and expected. It still is informative. What's not okay is that because 18S amplifies all eukaryotes, it also amplified Tar River spiny mussel, which I knew it would, and I included a primer blocking a muscle blocking primer. I apparently did not include enough of the muscle blocking primer because when I filtered out the muscle reads, my average reads per sample dropped from 20,000 to 2,600 reads per sample, and that is not a number that's high enough to capture the full diversity of the 18S gene. So I'm going to present this data, but it kind of has a huge asterisk on it, just knowing that I don't have everything that it could possibly be, 18S could sequence. So the first thing I did was look at, again, differences in the swab sites. I didn't find anything statistically significant, no difference between those sites. Again, that could be because I didn't capture enough of the diversity to capture the differences between the sites. But I did see, again, differences in all 4 of our propagated healthy, propagated unhealthy, wild healthy, wild unhealthy groups. And it at statistically significant values. So here's our 18S taxonomy bar plot. What I'll point out here is that those levels of diversity that we saw in 16S, we're not really picking up here. We don't see drastic level differences in the diversity between any of the groups. Caveat that I didn't pick up full diversity. So what did I pick up? So one thing that I picked up is Tetrahymena. This was seen only in the wild unhealthy group. This is a ciliate. It's associated with submerged or emergent vegetation that peaks in summer months. So this is interesting because the issues that we're seeing with muscle health also peak in summer months. So maybe this is what is being filtered in. As I understand it, the water is filtered in from outside ponds and then goes through the filtration system and into the muscle tanks. But maybe this is something that isn't being filtered out and is causing damage to our muscles. Similarly, only in the wild unhealthy, I see this Deoxystoma, which is another ciliate. This ciliate is a filter feeder and it feeds on bacterial biofilm. So this brings up an interesting question about if what we're seeing is pathogenic or opportunistic, because this Deoxystoma that we're seeing, we could be feeding on some kind of bacteria that is now growing on the wild muscles and it just likes to eat it. So it's not that that particular ciliate itself would be damaging to the muscle, but that it's could be feeding on something that's damaging to the muscle. I also saw in this group a couple of things that I only saw in our healthy animals. So this is a Spirogyra, it's green algae, and it was picked up in both of our healthy groups and not seen in our unhealthy groups. So this picks up another interesting question where we've been talking about trying to identify a pathogen, but maybe it's something that's missing rather than something that's present. Maybe these mussels need this green algae, and in the absence of the green algae, their health declines, which then allows them to be opportunistically colonized by bacteria that otherwise wouldn't be there. And then the last one that I'll mention also was found differentially abundant in our healthy groups compared to our unhealthy groups. It's all I can tell you is that it's a ciliate. So this highlights one of the challenges of working again with microorganisms in that a lot of these sequences that I'm pulling out don't have vouchered specimens. We don't know what they are. They're species that haven't been described. So all I know is that there's a ciliate there that is present only in our healthy groups and not in our unhealthy groups. And so with that, I will just take a minute to thank some collaborators and funding sources, Jeremy McCargo and Katie Patoka, who are my main collaborators on the American Shad Project, which is funded through the Federal Aid and Sport Fish Restoration Program. Rachel Hoke, who's my main collaborator on the Muscle Health Project, which was partially funded through U.S. Fish and Wildlife Service. And then also the North Carolina Museum of Natural Sciences, where I am based in the Genomics and Microbiology lab there, and where all of these samples were processed. So with that, I can take questions.
Thanks, Heather. That was a great presentation. I'll just ask everybody if you have any questions, please drop those in the chat. I'll start off just by asking you the perhaps the obvious question. You know, we have lots of different potential genetic projects across the agency, and with you being an agency-wide position, it might be useful for folks to know how best to approach you or request assistance if they have a project that they think has genetic elements in it.
Yeah, just the quickest way is just to send me an email, and then we can go from there and set up a meeting with any of the biologists or other staff that would be involved in that project. One thing to note is that I am the only conservation geneticist in the agency. I did get a technician recently, which is helping. But even if it's, so one of the things that we would discuss in the project is, is this something that we'll do within in-house at NCWRC, or if it's something that we want to try and contract out to another lab, because I just can't take on all the work that's going on. But I can help consult and find appropriate people and make sure the methodology is answering the questions that we want answered.
All right, that's great. Here's a question for you. Is there any way to get at whether the mussel health is from the presence of a microbe or the absence of a vital one?
Yeah, I mean, down the road, once we have identified what kind of bacteria or ciliates or whatever we want to test, we'll have to do additional testing on, well, what happens if we put it into a system, or what happens if can we filter it out of a system and see what happens. So the genetic standpoint right now is really identifying those things that might be of interest, and then we'll need to do some more studies where we're actually manipulating the presence or absence of that specimen within our tanks and seeing what's happened. But at this preliminary stage, no, but it kind of points us in the direction of where we want to be going for next steps.
All right, let's see. Can metabarcoding be used quantitatively, in other words, to get relative abundance of species?
There are some papers that have looked at that recently that do think it can be used. There's the caveat there is that you also, when you're doing the initial PCR, certain species will amplify better than others depending on how well those primers are matching that particular species. So you're going to get differential amplification in the initial stages, and so you have to be cognizant of that. But even still, the recent literature suggests that at least within one experiment where everything was sequenced together, you could look at relative abundance.
All right. There are no more questions in the chat. Unless somebody else has another one, I'll simply say there's lots of thanks for the presentation. I will echo that. Really good presentation, some great explanations about how the use of science in our conservation and management has and continues to expand. So I'll just say thanks again, Heather, for doing the presentation. Thanks to everybody for being here. And hope everybody has a good rest of your day.
Thank you. Thanks, everyone.